Hi guys, so today we will be starting with biomolecules. We did the introduction in the previous video, and today we will start with the first major macromolecule that we have, which is a carbohydrate, right? So as we all know, chemically speaking, if we are talking about the chemistry of carbohydrates, then they are hydrated carbons, right? So they are either aldoses, ketoses, or a fused form of them, condensation product of these compounds, right? If you look at the basic chemical formula for them, the empirical formula would go CnH2n whole n, where n is an integer and it can vary between 3 to 7. So it can be C3H2O whole 3. So that is C3A6O3. That's a possibility, right? But not above 7. The value of C cannot go beyond 7. You cannot have C8 in a carbohydrate, correct? Now, carbohydrates from the perspective of biology is extremely important because it is our primary energy giving nutrient, right? So it gives us energy. For that matter, the energy that flows through the ecosystem across the food webs is this carbohydrate. It is produced primarily via photosynthesis in green autotrophs, right? So when plants they do photosynthesis, they fix the carbon dioxide that we have in the atmosphere, convert it into glucose, and that's the primary carbohydrate that we have. For that matter, glucose is the primary carbohydrate for respiration in a very universal way, whether it's aerobic respiration, anaerobic respiration happening in a bacteria or happening in human, glucose serves as the basis for respiration, correct? So carbohydrates from the point of view of biology is an extremely important macromolecule. If we classify carbohydrates broadly, we can classify them on the basis of their structure or on the basis of the functional groups that they hold. They can have two functional groups. They can either be aldoses, where they will contain a CHO group, but the CHO group will always be terminal. Okay, so for the matter this one, this is a 1, 2, 3, 4, 5, and 6 carbon, so it's C6H12O6, which we know is the formula for glucose, correct? So glucose in this case will contain a aldose group at the terminal end of it, and such carbohydrates containing aldoses or the CHO group, right, containing aldehyde groups or CHO groups are called aldoses. Okay. The other group obviously contains a ketone group and they are called ketoses. For example, here, this is the ketone group, correct? So now, for the ketone group, it cannot be terminal, it has to be internal. It's inside the structure, it's not a terminal ketone group. In that case, we call it a ketose. This is fructose, for example. Okay, the examples that I have used in this on this board are the examples which are primarily important from the perspective of biology and from the perspective of boards as well as NEET. Okay, coming to the factor of classification, where we classify on the basis of size, or let's say the number of subunits that we have. So what do I mean by subunits? Again, if you look at the concept of macro and micromolecules, macromolecules tend to be a conjugation of many micromolecules. They are polymerized together, correct? So for example, if the bricks that build a house are your micromolecules, then the house finally becomes the macromolecule, right? So here, that is the brick, the subunit that I am talking about. So glucose, one molecule of glucose is the subunit. So singly, the single molecule of glucose then becomes a monosaccharide. So a monosaccharide has the basic empirical formula of CnH2nOn, which is this. Okay. Oligosaccharides are saccharides or carbohydrates which contain 2 to 10 subunits of monosaccharides. Okay. And anything higher than 10 subunits of monosaccharides, then it becomes a polysaccharide. If we want to talk about sugars, so if we are talking about sweetness, then remember this, that the smaller the carbohydrate, the sweeter it is. The more complex carbohydrates tend to become, the less sweet they also become, because then it gets harder for our the structure doesn't allow it to have that sweetness, and also digestion becomes harder. Our mouth, where we taste the sweetness, can only digest, because of the presence of tyaline or salivary amylase, the cooked starches or the cooked carbohydrates, correct? So therefore, that's why we feel it's sweet. The smaller a saccharide, a sugar, the sweeter it tends to be. So polysaccharides usually are not sweet, right? Polysaccharides, for example, starch, which you taste in, let's say, a raw wheat or raw grain of rice, they are tasteless. But think about fructose, for example, present in all fruits. It's sweet, right? But it's a monosaccharide. That's the whole concept of sweetness there. Coming back to the classification, if I look at monosaccharides, we can classify monosaccharides on the basis of the number of carbohydrates that they contain, I mean, number of carbons that they contain. So, in that case, if there are 3 carbons, then it's a triose, 4 then tetrose, 5 then pentose. This is an extremely important group of carbohydrates again, because the pentose sugars, they form the backbone of one of the most universal structures in biology, and that's your DNA and your RNA, that's your ribose sugar. Ribulose again is extremely important, because ribulose 6-phosphate and RUBP is actually one of the most abundant chemical substances found on planet Earth, because it is what is converted during photosynthesis in the plants, correct, for the C3 cycle. Coming to 6 carbon monosaccharides, you have hexoses. So your glucoses, fructoses, all of these are hexoses. And heptoses again contain, that's the final range, the 7 carbon monosaccharides. Okay, so this is on the basis of the number of carbon atoms. On the basis of the arrangement, if we wanted to classify, we could classify them as pyranose or furanose rings. These are ring structures, not linear structures, but ring structures. So the pyranose ring contains 5 carbons and 1 oxygen and has a hexagonal structure, whereas a furanose ring has a 4 carbon, 1 oxygen structure, and it's pentagonal in nature. The structures of the carbohydrates and also the anomalisms, they all fall under the scope of organic chemistry, and they would be explained to you in other videos. As of now, we will just focus on the biological aspect of it, right? Continuing to oligosaccharides. So oligosaccharides, if there are 2 units, then it's a disaccharide. If it's 3 units, then it's a trisaccharide. Okay. Now, 2 unit ones, again, very important one. Sucrose, stable sugar that we have. Maltose, that's again found in malt, right? Lactose, found in milk. Sucrose is made up of 1 fructose, 1 glucose. Maltose made up of 2 glucose. Lactose made up of 1 glucose, 1 galactose. So these are all disaccharides that we usually consume also in our daily diet. Raffinose is a trisaccharide which is made up of a glucose, a fructose, and a galactose. Okay. And polysaccharides are obviously more than 10 subunits of carbohydrates in each. Two very important ones are starch, which is the reserve food in plants, and glycogen, which is the reserve food in animals, in us as well. Polysaccharides are formed by a certain kind of bond, which is done in the formation of polysaccharides. It's called a glycosidic bond. Polysaccharides are basically the complex carbohydrates which are made up of more than 10 subunits of a monosaccharide. So we have two, starch and glycogen. We obviously have more, but starch and glycogen specifically are very, very important, because starch forms the reserve food in plants and glycogen is the reserve food in animals. It is formed by glycosidic bonds, which we will discuss in the next video, along with the other polysaccharide structures that we have. From now on, let's understand the concept of reducing sugars and the tests that we usually conduct to check for the presence of carbohydrate in a particular reducing sugars in a particular substrate. Okay, so reducing sugar is a concept where they reduce these sugars, they reduce the Cu2 plus ions into Cu plus ions. So you have a cupric to cuprous conversion, right? It's because of the reduction of the cupric into cuprous state. All of the aldoses and ketoses that we have, which are free in the carbohydrate structure, are all reducing sugars. So no matter whether it's a monosaccharide, all monosaccharides are reducing. Disaccharides also are reducing if they have a free aldose or a free ketose in them. However, sucrose is not reducing in nature because it is formed by the fusion of glucose and fructose, and the glycosidic bond is formed such that the aldehyde group and the ketone group are not, they don't stay functional anymore, and therefore it is not reducing in nature. However, maltoses, lactoses, galactoses are all again reducing in nature. So this is the aspect, the reducing nature of these sugars is what we test for in our reactions, and we establish the presence or the absence of the presence and the absence of carbohydrates in a particular solution, correct? There are two tests which are basically the similar other than the reagents or the compounds that we use. So we have a Benedict's test and a Feeling's test. Both of them use cupric sulfate as the basic compound which will be reduced into cuprous oxide, and we have other chelators which we will and alkaline substances which we use for the reaction to proceed. So in Benedict's test we use cupric sulfate with sodium carbonate and sodium citrate. When it is exposed to sugar, obviously the cupric sulfate, sodium carbonate, sodium citrate, which is the blue color, it forms a brick red precipitate of cuprous oxide, and that proves that it has been reduced. So the compound, the solution contained a reducing sugar. Fehling's test is similar, just the Fehling's reagent is different, where we have cupric sulfate, sodium hydroxide and sodium potassium tartrate. So in this, you get a brown red, brick red precipitation of cuprous oxide again, which again proves the presence of sugar. This is the basic overview and classification of carbohydrates that we have covered today. We'll obviously go ahead in the next videos, discuss glycosidic bonds and the other physiologically important monosaccharides, oligosaccharides and polysaccharides. So please keep following this series. Thank you.